We are moving on to new material, the endocrine system. We have a brief view of the endocrine system this week. Probably 2 classes on the endocrine system, which doesn't really do it justice, but we've had a lot of other material to cover so far in the class. It's not new in the sense that it's overwhelming because you've never seen something like this before. The endocrine system is a slower nervous system in a way. And this describes the system that releases and the actions of hormones. That's the endocrine system. And not just estrogen and testosterone, but also insulin as a hormone. We've talked about oxytocin way back when as a hormone. There are many different kinds, varieties, flavors of hormones, and there are many different kinds of responses. So we are discussing in brief the patterns of activity, how hormones work and are released, and we'll have one good example of a gland and the hormones that it releases on Thursday. So I'm going to try to parallel our discussion here with the nervous system. You'll be able to compare the nervous and hormonal signals or messengers, where the nervous system relies on action potentials and electrical signals. The the endocrine system relies on chemical messengers. We will describe the basic actions of hormones, describe where and how they're released, where they exert their effects, and we'll talk about the specifics related to those effects. There are 2 main classes of hormones, and they interact with the target cells very differently. Some of the highlights are the same, but the interaction is different. So we'll outline those 2 schematics, those 2 patterns, and we'll discuss how hormonal signals are modified. So you've got general activity, you've got 2 classes of interaction, and then there's layers of activity where a signal can be strengthened or blunted or might exert a completely different effect at the same target tissue. We'll talk about some of those mechanisms that modify the hormonal function as well. And then lastly, we'll explore in detail, hormone release at the pituitary gland. And it's one gland, it's a candidate example. There are a myriad of other glands. We just don't have time for all of them. But the pituitary as the master gland is a good candidate if we're going to pick one. So that's the flow through this week with review, remember, on Friday. So let's contrast these 2 systems, the nervous and the endocrine system. They both produce signals to affect some change. The difference, I guess, is in the type of change and the speed of the signals. They both act together to affect some change, and that means strengthening or lessening the activity of target cells in the body, to coordinate systems in the body. Whereas we know the nervous system releases neurotransmitters. Those are physical molecules like acetylcholine. We've seen those at least twice, if not more, in synapses between nerves at the neuromuscular junction. The endocrine system releases hormones. They're also molecules, and there are different types of molecules. So they do have messengers. The messengers are similar, but the ability to release those and the distance across which those are released changes. For instance, hormones will be released into the blood. Largely, the majority of hormones are released into the blood, and they circulate through the blood. And they're not really directed to a target tissue, as they are carried along by the blood, and if they run into a target tissue, so be it. They're directed insofar as blood flows through the veins of the body and the arteries of the body and carries the hormones with it. It's an effective system. Hormones do elicit effects at what we call target cells or target tissues, and we'll talk about that cascade coming up. But it's different from how the neurotransmitter signal is propagated. Action potential is carried throughout the nerves, across synapses, to the terminal end bulbs where they are released. They're not carried through the circulation. They are at the end of the terminal nerves ready to be released. So there's no guesswork about where they're released and if they're going to be released at the appropriate target. The nervous system is much more targeted. And I suppose it's true that they both regulate the activity of cells, but it's a simplified version on the nervous side. Regulating the activity was ligand-gated ion channels are activated. Membrane potential is affected, and then maybe the cell or the nerve depolarizes. On the nervous side, that's the extent of modifying the activity in the target cells. It's still activity, but it's specific. On the hormonal side, the endocrine system has a wider variety of effects, and those responses can be graded and intensified. They can change the permeability of membranes. They can institute proteins. They can activate genes and transcribe proteins. They can make it more likely for other hormones to work. The examples we'll get into, but the effects, there's a myriad more effects for hormonal responses at the target cells that are not just action potentials on or off. The differences are shown here briefly. In your textbook, you can review this table, but I think the most important of these are in the timing and the types of responses. So, nervous signals are the early warning signals. They are quick. They are generally short-lived. They're potent signals that don't last long. Incredibly fast transmission. Remember myelinated neurons, even versus unmyelinated, many-fold difference in speed of signal transmission. Both of those are far and away above how quickly a hormonal or endocrine signal can be transmitted in the body. Hormonal signals are slow. But hormonal signals can be maintained longer. They're more persistent. And so that's the distinction between acute and chronic effects of these two systems. The nervous system is acute, and the endocrine system is chronic, providing responses in the order of days to weeks even. And the types of responses. The types of target cells. The nervous system only activates excitable tissues, excitable cells, nerves and other muscles. The endocrine system can activate a whole host of different kinds of cells, not only nerve and muscle. The tissues are more broad, the actions are more broad, and we'll see how those broad responses are carried out shortly. Major difference in the speed and the duration of the signal transduction. Now, often these are integrated. Often the nervous system will activate a gland to secrete hormones, while at the same time activating a target tissue, so that there is a two-pronged approach to emphatically make a change. But we'll get into that. So, the endocrine system, releasing hormones, all due to the action of glands and or secretory cells. And if you remember way back in week 1, we introduced the idea of glands. And you saw glands in lab, and we identified there are different kinds of glands. And one kind are the exocrine glands, which simply means exo, outside of. The products are delivered outside of a tissue, either to the outside of the body or into a cavity of the body, but it's outside of a tissue within the body. And these glands are not the ones that secrete hormones. So these aren't the focus of this section. So we can cross them off the list. Sebaceous oil glands, sudoriferous sweat glands, those are not hormonal glands. They respond to hormones, but they don't release hormones. They are secretory, they have some of the same functions, but they're not hormonal. So, one important distinction. And then we move to our focus on other cells in the body that can release endocrine signals. The large majority are endocrine glands. And for lack of a better comparison, I think of these almost as sponges. And secretory cells are kind of like sponges as well, where the hormonal signals, the chemical messengers themselves are just, they're oozed out of the cell. And there's a better way to describe it. You'll learn about exocytosis, but they are delivered by the cell to the immediate environment. They don't have ducts. They are cells or a cluster of cells that secrete hormones directly into the environment surrounding those cells. And the environment surrounding any cell is the interstitial fluid. The space, the fluid between cells within a tissue. Now usually, these glands are highly vascularized. That is, they are in close proximity to arterial supply and capillaries. They're in very close proximity to the blood. So for all intents and purposes, if hormones are released into the interstitial fluid, they readily diffuse into the blood. There are exceptions to this rule, and I'll show you those. But the large majority will release hormones into the blood through diffusion. And hormones will be carried along by the blood until they reach their target cells and exert their influence. Now the glands, the major glands within the body that have large hormonal secreting abilities are listed here. The pituitary is what we'll focus on Thursday, which is a shame because the thyroid is also a very interesting gland, as is the adrenal, or are the adrenal glands. Really all of them are important, but maybe I'm biased towards a few. Thyroid, parathyroid, the pineal glands. You've seen a few of these, or heard referred to them as well. We're going to look at the pituitary only. The same processes apply to all glands, but the hormones released are different. And the locations in the body are different. Now, for cells to have endocrine activity, they don't only have to be a part of those glands. Some cells themselves can secrete chemical messengers, independent of being in a cluster of cells called a gland. I still consider these as part of the endocrine system because they are secretory cells that release chemical messengers that affect the local environment. But they are individual cells that do so, and these are cells of the pancreas that secrete insulin, cells in the stomach that secrete stomach acid that help with digestion, Some cells in the heart will actually release chemical messengers that modify whether the kidneys will reabsorb fluid. That's a wild departure from the normal function of the heart as we know it. But it's true. Atrial natriuretic peptide. The name's not important. So some secretory cells will release hormones that aren't necessarily part of glands. Glands will release hormones that are carried through the blood. Some of these cells will even modify hormones. We're getting a glimpse here at how complicated the layers of the endocrine system might be. So for instance, if we go back to the kidneys, hydration is one of the topics I study a lot. If you need to retain fluid, one of the chemicals that does that is angiotensin, and it's released by a gland near the kidneys, and it travels through the circulation, but it's not active in its original form. It has to go all the way through the heart to the lungs, where it's converted by lung tissue. It's flowing through the blood, it's converted by lung tissue into an active form, and then it circles back to the kidneys where it helps reabsorb fluid. So this is a complicated system with many layers of control that will allow you to understand perhaps how the response can be graded or intensified or finely tuned. And we can see a rough schematic of all of the secretory tissues, or the main secretory tissues in the body. The sum total of all of these secretory cells and glands I consider the endocrine system. And notice they are widespread. There's a widespread network of cells with endocrine function, which might provide redundancy or might provide layers of control. But look at how widespread these glands are. Thyroid and the larynx, the adrenal glands above the kidneys. We're going to look at the pituitary gland way up here. The ovaries, the testes, secretory cells. The stomach has secretory cells. Not all of them are highlighted. Widespread distribution throughout the body, allowing multiple layers of control and multiple sites of regulation. So this is for scope. Complex system, widespread, and now we drill down. If a cell or a gland releases a hormone, it has to do something. How does it accomplish its action? The large majority are endocrine hormones that pass through the circulation. When they travel through the circulation, they don't exert any specific effects until they find a cell with an appropriate receptor or encounter an appropriate receptor. Usually, these are membrane bound. That is, they're in the plasma membrane of a cell. So hormones have very specific effects. Hormones are the keys, and the receptors are the locks. And only one kind of key can open a certain type of lock. Only certain hormones can bind to and activate certain receptors. It's very specific. So we have hormones that can be released. They can be released in large quantities or small quantities. They travel through the body and maybe they find a target tissue. Then they bind a receptor. Even that's not static. We have layers of control at the receptor level as well. Receptors are just proteins, and like any protein in the cell, it's always being turned over. We're making new ones, breaking down old ones. We can increase the production of receptors. If we have a lot of locks, then it's very likely your key is going to find the appropriate trigger. Or we can regulate them so there are fewer receptors. And we do that actively to help control the response to some circulating hormones. The process of desensitizing, if you have a lot of hormones circulating for a long period of time, on the order of weeks to a month, the target cell will stop producing stop producing the receptor for that hormone to blunt that hormonal response. It's a way of negative feedback. Similarly, depending on the hormone and depending on the receptor, if there's supposed to be a certain amount of hormone in the blood and it's not apparent, target cells can increase the regulation and the production of receptors, making it more likely that if you even see a drop of that hormone, that signal will be conveyed. And this is long term. This is days to weeks or months. It takes a long time to make new proteins and put them in the membrane. So hormones, always being produced, always being broken down. So there's a balance. Receptors, always being produced, always being broken down. And individual hormones only fit into one type of receptor. This is a great example of structure driving function. Hormones fit into the receptors because they have the appropriate shape. The key is ground with the right teeth to open the lock. And so if something changes the shape of the receptor, all of a sudden that signal isn't conveyed as readily. So what we're describing through all of this is the main action of endocrine hormones shown here. The large majority act as circulating hormones. Most of the glands in the body function accordingly. As we described, they release their hormones. This is the sponge releasing hormones into the interstitial fluid, the white area. And then if those cells are in close proximity to the blood, like this one is, the hormones will diffuse across and travel as circulating hormones in the circulation. It's a bit redundant. And this will persist. This is the status quo until hormones diffuse out and find a target cell. They diffuse indiscriminately. They're not directed like neural signals are down specific branches of specific nerves. They diffuse through the bloodstream into the interstitial fluid of distal, faraway tissues. And if they happen to run into the receptor that matches the shape of the hormone, it'll exert some activity, change the activity of the cell or exert some effect. But if not, nothing happens. If that target receptor isn't there, the hormone doesn't do anything at that tissue. You need the hormone and the receptor, the key and the lock. So given that this is the pattern of activity of the majority of glands, we need two main things for the endocrine system to function normally, or for a signal to be transmitted. One, we need hormone release. And that should be pretty clear. I guess really we need three things. Two, we need the receptor at the target tissue. Three, we need blood flow. there is no way this signal will be transmitted without appropriate blood flow at the gland and at the target tissue. If there's not appropriate blood flow, the signal doesn't get where it needs to go. So that's often a missing link in considering the normal function of the endocrine system. Now, I did acknowledge there are exceptions to the rule. The majority of glands and hormones work as we just described. Those that don't are not circulating hormones. They're called local hormones. And it's perhaps best to think of most of the secretory cells in the body as having this type of function, as functioning as local release of local hormones. That's not universally true, but most secretory cells do this. Stomach cells secreting stomach acid does this. So hormones that don't enter the circulation are called local. And depending on their actions, whether they activate a different unique cell, they're called paracrine. And if they activate the same cell, if they self-regulate, they're autocrine hormones. The same things need to be present for this to work. You need release of hormone. You need receptors of the appropriate size and abundance. Blood flow is less of a concern, but you certainly need diffusion to the target tissue. But it's less of a roadblock here than it is with the circulating hormone example that we just saw. The difference is these do not enter the circulation. That's the big difference. Everything else still holds up. Lock and key, diffusion is required. Now, so far we've considered these small black dots to be hormones in general, a universal example of a hormone. But they are not all the same shape, size, they're not released in the same amounts, they don't have the same downstream activities, and in a broad sense, we can group them into two main categories. The type of hormone can be categorized in one of two ways. Either lipid-soluble, these are often synthesized from fats or cholesterols. They're made of lipids, and so they are lipid-soluble. You'll learn in biochemistry that these are non-polar, and that's not so important now, but because they're made of fats, they can dissolve in fat easily. And really what I mean is they can pass through the plasma membrane of cells easily. Plasma membranes are made of fatty acid derivatives. And so these can pass through the membrane of cells unopposed. But you all know oil and water don't mix. These are made of fat. They don't like water, and so they are hydrophobic. They don't dissolve in water. They don't dissolve in blood, which is mainly water. They don't dissolve in interstitial fluid, which is mainly water. They don't dissolve in cytoplasm, which is mainly water. So we have to make some concessions for these hormones to work in the body. The concession is shown here. They're bound to a chaperone. They're bound to a transport protein. It's like their personal bodyguard that takes them around within the body. They can move through membranes really easily. And so the opposite example, the other side of the coin, would be hormones that are water-soluble and lipid-insoluble. Water-soluble hormones are usually based on amino acids. They're made of proteins or the building blocks of proteins. They're not made of cholesterol and fat. So these can dissolve in the blood no problem. They don't need chaperones. They dissolve in the interstitial fluid, no problem. In the cytosol, no problem. But the plasma membrane might as well be a brick wall. They can't get through. And so these fundamental differences will drastically affect how each is able to register a change in activity at the target tissue. So, different classes of hormones, and within each class, many different types. Each type will have a different target receptor, and as such will have a different function. Myriad of possible outcomes that themselves can be changed. We already have a lot of potential outcomes, and the way in which we try to send that signal affects the outcome further. Even if it's the same hormone at the same target cell with the same receptor, there are a number of factors that can affect how a hormone works. And I'm just realizing what I'm describing to you is a slide that's coming up in 3 or 4 slides. So I'm jumping way ahead. That's still true. But the way that we can release hormones, the net effect based on how the response is modified could be, like we mentioned before, changing the permeability of a membrane, contraction of a muscle, activating new proteins, making mRNA, changing the location of things within the cell. All of those things, the wide variety of responses, are due to the wide variety of types of hormones and then how the signal is sent. So we're coming back to that in a second, after we discuss briefly how each class registers its effect at the target tissue. So first type, lipid-soluble hormones. Hydrophobic, so they don't like to dissolve in blood or the interstitial fluid or cytosol, but can move across membranes easily, and we see that here. These hormones don't bind to receptors on the surface of the cell because they can pass right through the surface. Oddly enough, their receptors, we think of them as satellites or antennas projecting out of something. The receptors are floating around inside the cell. So lipid-soluble hormones are carried by their chaperone to the target tissue, and then they just enter willingly. They're on the VIP list. No problem. They just get in and bind to a target receptor in the cell. They're chaperoned anywhere there's water, which is the majority of locations within the body. But they pass through membranes, they pass through tubules, they pass through membranes, they pass into the nucleus, which is also made largely of lipid byproducts, unimpeded. And it's this free access that allows these hormones to, I'm not going to say always, but one of their major roles is to bind to and transcribe DNA. They get right into the nucleus, initiate genetic transcription, and start to make new proteins. Anabolic steroid hormones are one example of this type of lipid-soluble hormone. And we don't know from this schematic what this new protein is or what it does. It could be a structural protein, actin and myosin. It could be a regulatory protein. It could be sodium potassium ATPase protein. It could go to the mitochondria. Massive, widespread downstream effects, depending on what kind of proteins are transcribed. Water-soluble proteins are more complicated. They can't get through the membrane, so they pass the message through the membrane. Water-soluble hormones are what we classically think of as hormones that bind to the antenna or the satellite dish on the surface of the cell. Water-soluble hormones bind to membrane-bound receptors. And they aren't taken up, usually. They can't just move through the membrane. Because they're there, their presence indicates that there's some important message that needs to be conveyed, some action needs to be taken, but they can't move into the cell to do it. They move freely in fluid but can't pass through the membrane, so they need a way to transmit the message. They need a way to signal the fact that they are there in abundance. And so in this case, the hormone acts as the primary or the first messenger. It's the first link in the chain of eventually bringing about a change in activity. Hormone is the first or primary messenger. It binds to a receptor, and usually what will happen is that receptor changes shape slightly. And that turns on something inside the cell and produces a second messenger. We're not going to go through the mechanics of how or what that signal is, but the second messenger is almost always this thing here called cyclic AMP. Cyclic AMP. Cyclic AMP is a derivative of ATP. Sending signals, initiating cascades, takes energy. We convert ATP into cyclic AMP. So the hormone isn't the thing responsible for a change in activity anymore. Now that it's initiated the production of cyclic AMP in the cell, this carries the message. It's taken the baton and it carries the message further into the cell. What that message is depends on the hormone. It depends on the downstream cascade of proteins that are activated. And often this also results in the transcription of new proteins and new genes. But overall, the activity is affected. The activity of this target cell changes in response to the hormone. So I'm purposefully not going into detail with all these numbered steps, because arguably that's the focus of biochemistry. But know that the signal, the message, moves from a hormone to a second messenger, and there might be 3, 4, 5 different signals or times that the signal is passed before the net result occurs. So now, the slide that I was trying to get to before, that I jumped ahead to, how does that signal change? How can we modify the signal? How can we make it more urgent to make new protein? Or how can we release more of some downstream hormone in a different gland or a different tissue? Right now, the description of our response is fairly linear. If you have a hormone, these steps occur. And if it's lipid-soluble or water-soluble, you follow a different recipe. But overall, something happens in the cell. But it's not on or off like that. The amount of change is dependent on the amount of hormone. If you've got an emergency situation and the pituitary gland releases a lot of one type of hormone, then the net effect at the target cell, the target tissue, is going to be magnified, right? More hormone, more binding, more change in activity. The signal can be changed or altered if there is more or less receptor. The cell can be more or less sensitive to the circulating hormones, more ready for that change in activity. Sometimes, whether the signal is transmitted depends on if other hormones are present. Depends on when other hormones are present. So if we think of the lock and key mechanism, imagine you have a deadbolt and then a handle lock. You're only going to open the door with your handle lock key if the deadbolt's also been opened. So sometimes a hormone can only activate and bind to the receptor if another signal is present. That gives us a lot of control over the types and the magnitudes of signals that we get on the endocrine side. Now, if those two work together, we call it a synergistic effect. They potentiate the response. They increase or enhance the response that you would get from one of them alone. Well, it makes sense if that is one pattern of activity, that the opposite would be true as well. Sometimes, if a second hormone is present, it prevents you from binding to the receptor. You can't put two keys into the same lock at once. Sometimes, a different hormone will bind to your ideal receptor and push the original hormone out of the way. Prevents any change in activity. We call that competitive inhibition. So many layers of modification that can intensify or reduce, blunt the effects of a hormone. And then the widespread downstream effects of the hormone are depending on the proteins that are transcribed and what the internal cascade of signals is. So there's no one recipe. All right, let's recap, and then we'll call it for the day. This didn't work last time, did it?
The endocrine system,
there we go,
along with the nervous system, controls body activities. In the nervous system, neurons provide rapid cell-to-cell communication at a distance with a short-lasting response. Most endocrine cells communicate slowly at a distance with long-lasting responses. Endocrine production cells communicate with target cells in the body using chemical signals called hormones. Most endocrine hormones are circulating hormones. These chemical signals travel through the bloodstream, facilitating communication between a production cell and a target cell. They pass from the secretory cells that discharge them into interstitial fluid and then into the blood. Only target cells with the appropriate hormone receptor will respond to the hormone signal. There are generally two chemical types of circulating hormones. Hydrophobic hormones, like estrogen, are also known as steroids and are derived from cholesterol. These hormones tend to be lipid-soluble. Hydrophilic hormones, like insulin, are based on amino acids, so they tend to be water-soluble. The chemical category, hydrophobic or hydrophilic, determines the method by which the hormone affects the target cell. Hydrophobic hormones are bound to a carrier protein as they travel through the bloodstream. These steroid hormones easily diffuse through the phospholipid bilayer of the target cell's membrane and bind with receptors within the nucleus. This binding forms a hormone receptor complex that directly affects gene expression. Targeted DNA sequences are then transcribed into mRNA. This results in the synthesis of new proteins. The new proteins are often enzymes which alter the target cell's activity. They cause cellular responses that are unique to that hormone. Everything from changing metabolic or contraction rates to the synthesis of other chemicals, including hormones, may be altered under the influence of a hydrophobic hormone. In contrast to hydrophobic hormones, hydrophilic hormones cannot pass through the target cell's membrane. These water-soluble hormones must bind to a receptor on the membrane. When a hydrophilic hormone binds to the hormone receptor, a G protein is activated. The activated G protein stimulates the membrane-bound enzyme adenylyl cyclase, which converts ATP into a second signal or messenger, known as cyclic AMP. Cyclic AMP acts to activate pre-existing cellular enzymes known as kinases. In a cascade of events, these enzymes either activate or inhibit other cellular proteins. While the action of the second messenger, cyclic AMP, is short-lived, the net result of the hormone signaling is an alteration of the target cell's physiological response. Even though the mechanism of action is different in water-soluble hormones, the range of physiological responses can be similar to those found in steroid hormones. The homeostasis of many of our body's controlled conditions, such as blood pressure, blood glucose levels, or metabolism, are maintained by the mechanisms of action of the hormones that the endocrine glands produce. These chemical signals are vital to our well-being.